Good morning, everybody, and welcome to day 2 of DDL. So I'm Ben Forbes. I'm a professor of pharmaceutics at King's College London. It's my great pleasure to introduce this first session of the morning, which is the Emerging Scientist Award. So it's an award we've been running for a few years now, and it's part of what we've been trying to do at DDL in terms of trying to support the people who come here. We've got the New Researchers Network, we've got our Career Development Awards. This afternoon we'll have the Young Investigator competition. And what we saw was there's a little bit of a gap in what we offer, in that we weren't really then having any progression as people progress through their careers. So we introduced the Emerging Scientist Award to recognise someone who's within 15 years of their PhD, but is really starting to do something in the field of inhalation science. So we ask for applications each year, and those applications are looked at by a panel of previous DDL lecture awardees, by the president of the Aerosol Society, and by a few members of the organising committee. And those applications are looked at for their relevance to inhalation science, to drug delivery to the lungs, for evidence of innovation, for scientific quality, for the cogency of the application and the promise that the work contains, and for the impact that it's starting to have. So we've been through that process this year, and we have a Emerging Scientist 2023, and I'm going to leave Regina to introduce that person.
So good morning also from my side. My name is Regina Schellies, and I'm a professor at Kiel University in Germany. And I have the pleasure to tell you who this year's Emerging Scientist awardee is. Maybe you can already guess it from the picture, because it's on there already. It's Olivia Merkel, and I may shortly introduce Olivia to you. Olivia is a professor of drug delivery at Munich University in Germany. She had been an assistant professor in pharmaceutics in the US for several years. She got her PhD in 2009, so you now can start calculating whether this is still within the 15 years it is. Her research focus is RNA formulation and pulmonary delivery for the treatment of a variety of lung diseases. So this is a nice follow-up also to yesterday's presentations. She got numerous awards and funding, and also got significant funding from the ERC. She recently also got, or was a co-founder of RNHale. So with this, I would like to invite you up on stage. Congratulations to this year's Emerging Scientist Award. So now she will present her work to you, and we will present the award to you afterwards.
Thank you. Well, thanks for awarding me with this prize this year. It's really a pleasure to be here, and I feel very honored, of course, to be this year's recipient of the award. You already heard that my research focuses on inhaled RNA delivery, and so, of course, this is what I will be talking about today. I unfortunately wasn't able to make it on time for the RNA talks yesterday because of the snow chaos in Munich, but I'll give you a short introduction, figuring that you probably heard some of this yesterday already. So one of the reasons why we are interested in inhaled RNA delivery is that there are many unmet medical needs. If you just look at the top 10 reasons of causes of death, according to the WHO, there are many pulmonary diseases. And particularly just coming out of this pandemic, we know, of course, how important it is to have better treatments for pulmonary or respiratory viral infections. But of course, also lung cancer, fibrosis, COPD, asthma, and many other diseases that target the lungs need better treatments. So while our solution or our approach to this is to make drugs for the currently undruggable, and of course with RNA, we have the chance to look into targets that so far have been considered undruggable, because with small molecules, we're often not able to interfere protein-protein interactions, but with RNA therapeutics, this actually gives us new opportunities. So you probably know that when we administer RNA formulations as they are approved currently, these are lipid nanoparticles. And no matter how we administer them to the body, they tend to accumulate in the liver. So particularly if you think of Onpattro, the first short interfering RNA-based therapeutic that was approved in 2018, that is also delivered with a lipid nanoparticle. It's injected and ends up in the liver, where it also has its target. But even when you were injected with the RNA vaccine a few years ago, about 6 hours after your injection, if you had checked for biodistribution, you would have found a lot of this RNA in your liver. So we know that with the currently approved administration routes for injection, we unfortunately can only target the liver. And so our idea of trying to deliver beyond the liver, and particularly to the lungs, of course, moves into inhalation science. But unfortunately, if you look at the currently approved formulations for RNA, these are, as I already mentioned, lipid nanoparticles that don't withhold the shear force and also temperature force during the process of nebulization. So unfortunately, nebulization is oftentimes difficult for delivering these LNPs and also maintaining colloidal stability and keeping an eye on quality parameters of these LNPs. So our solution to this is to develop different nanocarriers, and these nanocarriers that we develop are based on biodegradable polymers. Now, the way these new nanocarriers are often designed is by synthesizing large libraries of materials, and then often finding that only one or two of the candidates actually are really efficient in encapsulating, delivering, and also delivering into the cytoplasm, where we have the site of action of RNA. So our new approach, and something that I think we will see a lot more in the coming years, is to develop such nanocarriers, no matter if polymer-based or lipid-based, by artificial intelligence and by making more rational and also simulation-based decisions. And this is what I want to talk about today. Now, in terms of pulmonary drug delivery, I'm certain that I don't have to explain to this audience what the advantages are. But I think one thing that we need to keep in mind, particularly for nanocarriers in the lungs, is that we also have a few barriers. So if you think of mucus present in the upper airways and then surfactant in the lower airways, surfactant is a significant barrier for the stability of lipid-based nanocarriers. And something that we need to keep in mind for RNA therapeutics is that, as I mentioned before, we need to deliver them to the cytoplasm of cells. So what you see here is an example of short interfering RNA. Short interfering RNA usually is delivered in order to downregulate gene expression. So this is something that you can use, for example, against genes that don't belong to the body. So if you think of respiratory viral infections, for example, but also for overexpressed, pathologically overexpressed genes or oncogenes that with a short interfering RNA you can downregulate. Usually, if you deliver messenger RNA, you try to increase the expression or to deliver something that the body endogenously does not express. But again, the target is in the cytoplasm. So as you all certainly know, RNA is a large macromolecule, negatively charged, which doesn't enter the cell by diffusion. So what we need to do in order to protect the RNA and also to trigger uptake is to encapsulate it. For short RNA, we can sometimes also make covalent conjugates, where we can trigger endocytosis by receptor-mediated interactions. But particularly for large RNAs, it's important to encapsulate and protect it. So you see various RNA molecules encapsulated into a nanoparticle that then interacts with the cell membrane to be taken up by endocytosis. And then we usually encounter the next big barrier. And this is the so-called endosomal release. So a nanoparticle that is taken up by endocytosis initially ends up in the endosome, and it needs to escape from this endosome in order to release the therapeutic RNA into the cytoplasm. Because no matter what RNA we deliver, if it's short interfering RNA that needs to be incorporated into the so-called RNA-induced silencing complex, or messenger RNA that needs to interact with the ribosome, we need to release it from the endosome and also from the particle in order for its therapeutic action to happen. So for this short interfering RNA, it usually binds to base complementary messenger RNA and then activates the endonuclease activity of the silencing complex in order for the messenger RNA to be cut and be degraded. And with the message degraded and cut, we lose the message, so the gene can no longer be expressed, the protein can no longer be produced. So this is the basis of a lot of the projects that we focused on. And one project very dear to my heart is our asthma project that we've started many years ago, where the focus was in T-cell targeting. If you've ever tried to transfect T cells, if you've ever tried to deliver nucleic acids to T cells, you probably know that these are notoriously difficult to transfect cells because they're protected. They're our immune cells. They don't easily endocytose materials. And what we found out, however, is that when T cells are activated, so during a process of inflammation, they upregulate a receptor that's called CD71. And CD71 is nothing other than the transferrin receptor. So if you are in drug delivery, you might know that transferrin receptor targeting has been used for many years in oncology, but it hasn't really been used in inflammatory diseases. So if you look at the role that T cells play in asthma, and this is a very busy immunology slide, so I don't expect you to understand all of the details, but looking at the role of T cells, you'll see that they play a very central role. And they secrete a lot of cytokines that then activate downstream cascades and pathways that then cause all of the symptoms of asthma. So it's essentially like a conductor of an orchestra of inflammation. And one idea is to downregulate and interfere with the Th2 transcription factor GATA3. So if you downregulate GATA3, the idea is that you can downregulate all of these cytokines that are downstream of GATA3. Because if you only downregulate one cytokine, you will usually see an upregulation of the other cytokines. So the idea of silencing the conductor of the orchestra will then silence the entire orchestra. But for that, you need to deliver the RNA into the T cells. And again, this is usually a pretty big challenge that viruses are very good at, if you think of HIV, for example. But we also luckily managed to develop a bioconjugate of a polymer that's called polyethylenamine with transferrin to encapsulate RNA and to very efficiently deliver this artificial virus into T cells. And we tested this in our in vivo asthma protocol, where we used an ovalbumin model, where ovalbumin is a model allergen that we can sensitize mice against, and then we challenged them intranasally. So you can see how the mice are sitting in these inhalation tubes. And since they're obligate nose breathers, they will inhale the allergen. We just recently bought the new equipment, and we're very happy that we're now able to repeat a lot of this work. And what we found at the time is that if we deliver a fluorescently labeled RNA, and we make single cell suspensions after the treatment period, we find a lot of this RNA in the T cells of the animals with all the symptoms of asthma. But in our control group, the sodium chloride group, where animals were not sensitized and did not have lung inflammation, we didn't see any uptake. So we figured that this is really a receptor-mediated process that we also confirmed ex vivo in primary T cells. But of course, it was also important for us to show that in vivo, in a setting in the lungs, where we have cells, a lot of different cells other than just isolated T cells, we can really target T cells and get specific uptake and targeting of these T cells. So this was right around the time I moved to Germany, and you might know that getting animal protocols approved in Germany can take very, very long. So we started looking into 3R models. And I think this is also something that we will see a lot more in the coming years, also considering that now the FDA considers 3R models and more than they used in the past. And something that we really enjoy doing is using human lung tissues. So we have ethics approval to use peri-tumor tissue that we obtain from the different hospitals in Munich and Gauting. And we can fill these tissues with agarose and then cut them into very thin vibrotome slices. And then we usually don't get the tumor tissue. That's what pathology needs, of course, but we get the peri-tumor tissue. And the beauty of this model is that it still contains all cell types. So it contains, of course, the epithelium, but it also contains all the mobile cells, including T cells, dendritic cells, macrophages, and so on. So we can activate the T cells in this tissue just like we usually do ex vivo with dynabeads, for example. And then we can perform our transfection like we would do in vivo or also ex vivo with the primary T cells. And then we can perform many different analyses, including also toxicity experiments with human tissue, which is quite impactful to understanding the potential side effects of a treatment. So in these results, you see that we indeed managed to downregulate GATA3 expression in this human lung tissue very efficiently with our different nanocarriers. And since we're very interested in moving this towards the clinics, we also looked into a more clinically relevant administration route. So in our animal models, we usually intubate and we administer a nanoparticle suspension, which obviously is not very clinically relevant. But we also early on looked into spray drying of these materials in order to obtain dry powders for inhalation. And what we do is that we use our nanoparticle suspension, which we dilute in a sugar or sugar alcohol solution. And then we spray dry with the regular Büchi spray dryer in order to obtain what we call a nano and micro particle. Because what you see here are microparticles with a geometric geometric size of about 3 microns. But it consists of many, many nanoparticles that are glued together into this sugar matrix. And the idea is that now we obtain the right aerodynamic diameters and the right aerodynamic properties for inhalation, ideally even into the deep lungs. But of course, in order for these nanoparticles to be endocytosed, they need to dissolve into nanoparticles again. So these microparticles would not be able to make it into the target cells. And so it took us quite long to optimize the spray drying conditions and many of the technical parameters, but we managed to come up with procedures where we can now maintain colloidal stability and redispersibility of these formulations in order to show that now also the spray dried materials indeed lead to a significant downregulation of GATA3 in our asthma model. So while we were still doing these experiments, the pandemic hit, and I think probably everyone in this audience also looked into how can we do something against this awful virus infection, this pandemic. And we started to work with animal models, and you probably know that the ACE2 expressing mouse model was not necessarily an ideal animal model. The animals that we treated often died from encephalitis more than from their lung symptoms. And so we figured that knowing how well human lung tissue can be infected with the virus, we might go back to our ex vivo human tissue. So we used ex vivo human tissue and infected with wild-type virus in collaboration with a group in virology, and started with a prophylactic setup where we pre-treated the human tissue with our nanoparticles with short interfering RNA that the virology group had optimized, and then infected with a wild-type virus in order to check 24 hours later if we were able to halt the viral infection or prevent it. So our virology collaborators looked into different siRNA sequences and modifications, and based on different experiments, also using a GFP expressing viral model, they were able to show that with these sequences, with various sequences and modifications, they can decrease viral replication efficiently. But when we used the ex vivo human tissue, we were able to show that with our polymer nanoparticles and their best performing siRNA, we were able to downregulate genomic RNA very efficiently. But everything that I told you about now was done with non-biodegradable polyethyleneamine. And as I mentioned before, we're very interested in biodegradable materials that would have the advantage that they should be tolerated better by humans, of course. So many years ago, we started looking into spermine-based materials because spermine is a polyamine that in sperm, for example, very efficiently condenses DNA into the very small size of a sperm. And what we found is that spermine by itself, or even a bispermine or a tetraspermine, is still not efficient enough to encapsulate this very rigid and short double-stranded RNA. So our experiments showed there was some uptake in cells and cell culture, but we weren't quite happy with these results. So in the last couple of years, we focused on making polyspermines with a polybeta amino ester backbone, which is very simple chemistry and things that also we as pharmacists can do. And we have developed different materials that have a cationic spermine side chain and also a hydrophobic oleolamine side chain to obtain materials that are more or less amphiphilic. And so with these materials and with these monomers, we can play around to obtain materials with different ratios of cationic to hydrophobic side chains. So here on the left, you see the most cationic materials with 90% spermine, and then towards the right, it becomes more hydrophobic and more amphiphilic with 50-50 of these two different side chains. We can synthesize them in different molecular weights, different sizes. And with these materials, we started screening how well they can encapsulate RNA into nanoparticles of about 100 nanometers or smaller, with ideally a slightly positive zeta potential depending on the composition of the polymers. And then we checked for uptake into cells, lung cancer cells in these experiments, to see that the most cationic ones are taken up very efficiently, but this endosomal escape problem that I mentioned before is not overcome by these materials. Because if you look into this gene silencing efficacy here, where we always compare a negative control siRNA with the actual silencing RNA, you see there are no differences. So we went on testing the different combinations of materials, and we saw that the more hydrophobic materials, they weren't taken up as efficiently because we lacked the cationic charge for interaction with the cells. But the little material that made it into the cell also was released from the endosome quite efficiently, so that the gene silencing worked well. And now if you look at the 50-50 polymer with the highest hydrophobicity, you see that the uptake was amazing. So if you compare this to a 25 kilodalton PEI, it was really mind-blowing to the students that did the work. And most of all, we get about 96% gene silencing in vitro, which they were even more happy about, of course. So we went on with more relevant models. The work that I just showed was done in 2D cell culture, but of course we know that doesn't reflect the lungs, the situation in the lungs very well. So in this air-liquid interface model, where we have mucus shown in green here as a strong barrier, we didn't obtain the 96% gene silencing, obviously, but with one of the formulations, we had about 50% gene silencing, which is comparable to lipofectamine, a commercially available transfection reagent, which is very toxic, however. So my students went on, and with a previous generation of these materials, they also looked into different applications, such as mutated KRAS, which is a big problem in lung cancer, for example, and managed to compare two different types of BOC-protected, slightly hydrophobic, and deep-protected materials to show downregulation of this mutated KRAS, which resulted in a decrease of motility and invasiveness of these lung cancer cells, shown here in this Boyden chamber assay. And what I wanted to talk about today is that, as I mentioned before, we're now also trying to involve more simulation work and predictions into our development of materials. So going back to these oleulamine, spermine copolymers, we use a lot of molecular dynamics simulations in order to understand materials before we synthesize them, because it helps us to decide if this is a material worth synthesizing or not, and it also improves our sustainability in terms of wet lab work. So what you see here is the 10% oleulamine, most cationic material that forms quite nice particles, but you also see some of these what we call unproductive particles, because they don't encapsulate RNA. So they form particles, they form micelles, but don't encapsulate RNA. With the 50-50 material, it looks much better. There are very few of these unproductive particles, and encapsulation efficiency is very good. But if we look into 80% oleulamine, 20% spermine, we seem to see aggregation. And so this is something that, of course, we still have to confirm with real-life experimental work. But it really helps us to get a better handle on formulation development. So this is why I mentioned that in our current work, we use artificial intelligence, but we still synthesize small materials, small libraries of materials, check for their in vitro performance, in vivo performance, but also include the molecular dynamics work, and also incorporate something that's called molecular descriptors to describe the chemistry and the properties of the materials we synthesize. To then fit all of this into a computational model to train a machine learning algorithm in order to predict better materials, and then go back to the lab, synthesize these materials, perform all these experiments again, and see if we can validate our model or if we have to actually adjust the machine learning model. So the last part that I wanted to talk about today is still polyspermines. And these are polyspermines based on a polyacrylamide backbone. And the idea is still very, very similar. So here we have a decylamine rather than an oleulamine. And we also have spermine as the second side chain. And so again, with different ratios of these two different side chains, we see very good encapsulation efficiency. Here a little bit more cationic zeta potentials, very good encapsulation of the RNA at a very low excess of a polymer over RNA. And here, most importantly, we see that these materials very efficiently deliver RNA in an air-liquid interface model. So these materials are particularly good for delivery in the presence of mucus. And now you see that we again achieved about an 80% gene silencing in air-liquid interface, which is much better than lipofectamine, considering that lipid materials tend to get stuck in mucus. So we went on with these experiments, checked for the in vivo distribution within the different lung cells, and found that most of the uptake happens in type 2 pneumocytes, more so than in macrophages, which is great because macrophages, of course, tend to phagocytose nanomaterials. And therefore, we went on now with patient material from patients that suffer from lung fibrosis. So these are usually transplant lungs, where we obtain tissue from these patients. And we were able to downregulate PAR2, which is an emerging target in lung fibrosis, more efficiently with our materials than with the commercially available transfection reagents if you compare these two bands. But even more importantly, and this was done in 3 different patients where you see, of course, some variation, but most importantly, in these ex vivo lung tissue, we're even able to downregulate collagen production very efficiently. Looking at these 2 donors, we almost saw a complete disruption of collagen production, whereas in this one donor, of course, with a variety of donors, not all patients benefit the same. So in this video, you can hopefully see our RNA distribution shown in red. Blue shows the nuclei stained, and yellow is the autofluorescence from the fibrotic tissue. So here in this video, you can hopefully appreciate that when we deliver our RNA with these polyacrylamide-based materials, we can deliver RNA essentially into every single cell of these lung tissue explants. And therefore are very much looking forward into treating mice in a bleomycin model with these materials. So with this, I'd like to summarize what I've been presenting today. So I hope I was able to show you the potential we see in therapeutic RNA delivery to the lungs for treating respiratory diseases in the different in vitro, ex vivo, and in vivo models we apply. And most of all, I think as an outlook for future and where this field is going, I think that we will see more and more artificial intelligence work also in drug formulation. Of course, a lot of simulation-supported research, and then, of course, also the use of more and more 3R models before we actually move on into animal experiments, particularly with a focus on respiratory viral infections, where the pandemic has shown us that we're quite weak at the moment, with our overall aim of developing nanoparticle-based nucleic acid therapeutics. And with this, I'd like to thank the people in my group. Without them, I would not be here because they are the ones that actually do all of the work. You can see Min and Siyu who are here presenting posters. It's also Min's birthday, so happy birthday, Min. And I'm really happy to have such amazing people in my lab. Thank you.